Alright, today we are going to start about hypersensitivity type 2. It's very easy. Hypersensitivity type 2 is an antibody mediated. It refers to as cytotoxic because it triggers certain reactions which damages our cells. The antigens involved in hypersensitivity type 2 may be intrinsic, like in case of autoimmune reactions, or it may be extrinsic antigen, like maybe a part of a drug when absorbed into a cell that looks as an antigen to our immune system. Remember that antigens in hypersensitivity type 2 will always reside on the cell surface, like maybe on RBC, the membrane of red blood cell, or it can be an extracellular matrix, but on a basement membrane. These are the antigens that are involved in hypersensitivity type 2 that is going to bind with antibody activating the complementary system, and then resulting in different disorders and diseases. One of the antigen, like maybe quinine, which results into thrombocytopenia, or penicillin, which leads to hemolysis. These are the antigens when bind to the antibodies will activate the complementary system. How does it do it? We'll study in next slides. But these antigens are responsible for disorders and diseases in our body, the same like others. Now, what are antibodies, and what type of antibodies are in hypersensitivity type 2? So remember, in hypersensitivity type 2, we have two immunoglobulins antibodies that are responsible for the activation of complementary system. IgG, which is a monomer, and IgM, which is a pentamer. These antibodies are produced by the B cells, which is a class switcher, which means that it switches the classes of antibodies, like from IgG to IgE and from IgE to IgG and IgM, which is a plasma cell. Now, before knowing the exact mechanism, it is important to know the structure of antibody. Each antibody is made up of 4 polypeptide chains, 2 light chains, which is a binding site for antigen, which is known as Fab region, and the heavy chain, which is actually responsible for the activation of the complementary system, which is known as Fc region. As you can see right here in this diagram, this is the structure of antibodies, consisting of Fab region and Fc region. The Fab region is responsible for the binding with the antigen present on the cell surface, and Fc region is responsible for the system. Now, hypersensitivity can be of two types. It can be cytotoxic or it can be not cytotoxic. Cytotoxic means that due to immune reactions, it is going to destroy the cells or tissue damage. And non-cytotoxic, which means that it will bring alteration in the normal functioning of our biological system, maybe in organ. Before knowing how actually this works, let us meet some actors that play a definite role in hypersensitivity type 2, the complementary system. Remember, this complementary system was actually activated by the Fc region when antibody was binded with the antigen on the cell surface with its Fab region. Complementary systems are the series of proteins that are produced by the liver into the blood circulation. When they are in the blood circulation, so they are not active, they don't perform actions usually. And there are more than 20 proteins that interact with each other responsible for doing different things and mechanisms. They are only activated at the time of inflammation and when there is an immune reaction, like for example C3, when cleaved into C3A and C3B. Now they are the active forms of C3. Let's consider as an example penicillin. When some part of the penicillin, which is a drug, antibiotic, usually help our body to fight against bacteria. When a part of the penicillin is absorbed on the cell surface, now this part of penicillin looks as an antigen to our immune system. Antibodies will be released from the blood circulation, and antibody from its Fab region will bind to the antigen. Now, this binding is going to have some reactions, and it will trigger certain processes that's going to lead to certain disorders and diseases. It's going to release inflammatory mediators, It's going to promote the opsonization and going to lead the formation of the membrane-attack complex known as MAC and SHART, and also the release of the neutrophils, which is the natural killer cells. As a result, the cell is going to die. This leads to the lysis of the cell. For example, we talked about RBC, so it's known as hemolysis. The classical pathway of the complementary system. How it's actually activated. As I told you earlier, that these complex series of proteins are activated by the Fc region of antibodies. Usually, the complementary proteins are inactive in circulation, but when cleaved, they become active. Now, this is very important to know that the first valuable product that is formed after the activation of complementary system is C3 convertase, which is an enzyme. Now, this C3 convertase is going to activate C3, and then C3 is going to be cleaved into C3A and C3B, which is the active form of the complementary. They both separately do different things. Now, C3A with C5A, they are responsible for the inflammation. They are the inflammatory mediators. Wait for C5A, we're going to study it in the next slide. C3b is an opsonizer, which means that it's going to enhance the process of phagocytosis. Let's study C3b first. C3b opsonization, first of all, it means that it enhances the ability of an antigen to be phagocytosed, which means that engulfed and destroyed easily by the macrophages. Like, for example, in case of red blood cell, when an antigen is on the RBC cell surface with C3b opsonin, which is activated by the antibody from Fc region on its surface, it will be very easily recognized by the macrophages and neutrophils, which will lead to phagocytosis, means hemolysis. As right here you can see a macrophage going to engulf a cell, for example a bacteria having antigen on its surface, and the macrophage having the complement receptors, like for example the receptor for the C3b opsonin present on the cell surface of the bacteria. So it will be easily recognized by the macrophages. Now, C3b can also bind to C3 convertase. When C3b binds with C3 convertase, it will lead to form C5 convertase, and this C5 convertase is responsible for the activation of C5, which will be cleaved into C5a and C5b, which are the active form of C5. As we discussed earlier in the previous slide, that C3a and C5a are the inflammatory mediators which promote the vasodilation by the vesicular permeability, and is also responsible for the chemoattractants that occurs intracellularly. The chemoattractants are actually responsible for the attraction or chemotaxis of the neutrophils and monocytes for the phagocytosis. And this is with the help of activation of mast cells. When mast cells are activated, they release certain chemoattractants, and then they will attract the neutrophils and monocytes. C5b actually initiates formation of MAC. We knew that C3b and C3 is also actually responsible for the formation of C5 convertase, and then C5 convertase is responsible for C5 and then cleaved into C5a and C5b. So C5a was inflammatory mediator. Now C5b initiates the formation of MAC, which is a membrane-attack complex. Now what happens here is that C5b binds with C6, C7 and C8, which is forming a complex protein, a large protein, which is C9. Now this C9 is actually a MAC, membrane-attack complex. And what does it do is it's actually a transmembrane channel which is formed on the cell surface. Now These channels actually disrupt the homeostasis of the cell membrane, which is leading to the diffusion of free molecules in and out of the cells, and it leads to the lysis of the cell. Here you can see it. It's a transmembrane channel which is formed with the help of C5b with binding with C6, C7 and C8, and then forming a complex protein which is C9 and allowing the in and out of the molecule very freely. Now coming to the actual disorder and diseases, that how this is actually happening in hypersensitivity type 2. Like thrombocytopenia, for example, when quinine acts as an antigen, it attaches on the surface of platelets. Now what happens is that it results in binding of the IgG and IgM antibodies to the quinine, which is an antigen, and then it leads to the activation of complementary system and hence causes lysis of platelets. As we discussed, that antigen can be bacteria too. So antibodies form against the M portion of Streptococcus pyogenes. It leads to the activation of complementary system, and then it causes cross-reaction with the cardiac tissues, which damages the heart valves. Hemolytic anemia. In this case, maybe a particle of penicillin can act as an antigen, which results in the binding of antibodies and then activating complementary system, which leads to the hemolytic anemia. As you can see right here, and a part of the penicillin when attached to the cell surface of RBC, so it will lead to the binding of IgG antibodies with the antigen and then activating the complementary system, and then that complementary system is responsible for the hemolytic anemia, lysis of the cell. So thank you so much, friends. Hope you enjoyed the videos. Don't forget to subscribe to the channel.